Thank you very much for giving me this opportunity to share our experience for the spectrum of NTRK fusion-associated pediatric tumors in the Division of Genomic Diagnostics in Children's Hospital of Philadelphia. Sorry. How do I move to the next slide? This is it? For this one? Okay. So neurotrophic tyrosine kinase genes, NTRK1, NTRK2, and NTRK3, encode members of the tyrosine receptor kinase protein family TRKA, TRKB, and TRKC, respectively. Genomic rearrangements in the NTRK genes lead to TRK fusion proteins that are involved in different pathways, including PI3K, RAS-MAPK, and PKC pathways that are critical for tumor cell proliferation, survival, invasion, and angiogenesis. NTRK gene fusions act as oncogenic drivers of various tumors. The picture on the left gives you examples of the previously reported 5' gene partners of different NTRK fusions with different dimerization mechanisms that are either lead to constitutive activation of the tyrosine kinase domain or the overexpression of the fusion protein. So the TRK kinase domain is always included in the oncogenic fusion protein. But the transmembrane domain is not always present. Previous literature has showed that they might have an effect on cellular localization of the fusion protein. So NTRK fusions can be found in both adult and pediatric cancers. They are usually observed at a very high frequency in specific rare tumors, depicted in red here, with a frequency as high as 90% in specific patient cohorts. However, they are usually observed as a very low frequency in most common cancer types, including lung cancer, breast cancer, and melanomas. The annual incidence of NTRK fusion-driven tumors is estimated to be 1,500 to 5,000 cases in the United States. So since the identification of the first NTRK fusions in 1980s, we have gained more knowledge about the structure and function about these NTRK fusions. These lead to the development of TRK inhibitors, which could result in tumor type agnostic responses in NTRK fusion-positive cancers. Recently, FDA have approved larotrectinib and entrectinib as treatment for adult and pediatric patients with NTRK fusion-positive solid tumors. So in the DGD, we offer a fusion panel which interrogates 110 major fusion partners associated with over 600 known cancer fusions and many novel fusions. And this is used the Archer Anchor Multiplex PCR technology, which uses the targeted specific unidirectional primers, along with the universal reverse primers that could detect the gene target of interest, along with the known or novel fusion partners. Since 2016, we have tested over 1,400 cases from 1,150 patients. And NTRK fusions were detected in 25 tumors from 22 patients, which is about 1.6% of all the tumors has tested. So this figure on the left have showed you all the previous NTRK fusions that we have reported in the DGD. As you can see, ETV6 NTRK3 has the highest frequency in our patient cohort, which is consistent with the previous literature has shown. And we also have observed a wide range of different tumor types, including AML, different sarcomas, brain tumors, and some rare tumor types. And interestingly, we have observed that even with the same fusion, we can see they have been found in different tumor types, such as this RBPMS NTRK3 fusion that we have observed in both spindle cell sarcoma and T lymphoblastic lymphoma. In total, 10 out of 25 fusions that we have observed were either novel or rarely reported in the literature for other tumor types. And in 19 patients, the fusions identified have a confirmed clinical pathological diagnosis. And in 3 patients, the diagnoses were changed due to the identification of NTRK fusion. So in the next few slides, I will give you a couple examples of how the identification of an NTRK fusion can significantly impact the tumor diagnosis and the treatment options. Patient number 1 is a 3-year-old male reported right-sided preauricular neck mass in 2016. His MRI findings were concerning for a primary neoplasm of the right parotid with atypical imaging appearance, suspecting a mucoepidermoid carcinoma. And then he underwent a right superficial parotidectomy with resection of a parotid mass with facial nerve dissection. His molecular findings is significant for a fusion gene, ETV6 and NTRK3. And we also have found some non-diagnostic sequencing variants, including the sequencing variant in FANCC and multiple copy number variants. So this image showed you the identification of ETV6 and NTRK3 in our analysis software. And the gray bar here showed you the read depths that's supporting this fusion. And the blue and the red bars showed you the representative reads that encompass the breakpoint between ETV6 and NTRK3. And the bottom panel have showed you the breakpoint is between ETV6 exon 5 and NTRK3 exon 15, which contains the intact protein kinase domain. So what is ETV6 and NTRK3? It is a common genetic alteration occurring in several different tumor types, including congenital fibrosarcoma, secretory ductal carcinoma of the breast, papillary thyroid carcinomas, and some other tumor types. And the picture on the right showed you the common gene fusions that are found in different salivary gland tumors. And ETV6 and NTRK3 is diagnostic of memory analog secretory carcinoma in the context of parotid gland carcinoma. And so because of the presence of this gene fusion, the diagnosis of this patient has been changed from the mucoepidermoid carcinoma to MASC. So MASC is a low-grade malignant tumor of salivary gland with usually good prognosis. However, accelerated clinical course and high-grade transformation could result in poor outcome. So with this diagnosis, this patient has underwent a modified neck dissection, and his latest MRI has showed no evidence of a recurrent or metastatic disease. However, should the patient relapse, knock on the woods, he won't, the NTRK inhibitors could be considered as one of the treatment options. So patient 2 is a 6-month-old male with no significant birth or medical history. He was presented to CHOP for evaluation of a left upper extremity mass, and his histology was suggestive of intermediate to high-grade mesenchymal neoplasm. But the differential diagnosis, including vascular lesions, infantile fibrosarcoma, leiomyoma, and rhabdomyosarcoma. His FISH analysis for detection of translocation associated with ETV6, however, is negative. So this is his MRI imaging when he first presented to CHOP with the tumor here. And after a month, you can see the size of the tumor has been increased. So he came to us, and we found the molecular findings is significant for a novel fusion, which is this SPECC1L and NTRK3 fusion, and also with multiple copy number variants that are non-diagnostic. So this is the identification of this fusion on our analysis software. And because this is a novel fusion, we have confirmed it using nested PCR and Sankar sequencing. So what is SPECC1L NTRK3? So SPECC1L encodes a coid-coid domain-containing protein. The encoded protein may play a critical role in actin cytoskeletal reorganization during facial morphogenesis. And the identification of this fusion is consistent with the diagnosis of infantile fibrosarcoma. IFS is a malignant fibroblastic tumor commonly found in children under age of 1. And it has an incidence of 1% of all childhood malignancies with usually favorable prognosis and less likelihood of metastasis. So the treatment usually involves surgical resection, but chemotherapy and radiotherapy are usually used when complete resection is impossible. And misdiagnosis can lead to delay in treatment and also suboptimal outcome. So with this diagnosis, patient number 2 received an overall treatment plan for 3 cycles of larotrectinib, and then decide whether there's a need for surgery with adjuvant larotrectinib. And so far, the patient has been tolerating it very well, and his latest clinical exam showed a significant decrease in the size of the tumor. As you can see here, this was the MRI imaging showed that was taken a couple weeks ago with this patient. So in summary, we have identified NTRK fusions in 1.6% of all the tumors, which is about 3% in solid tumors in DGD tested, including the PTCs, soft tissue sarcomas, brain tumors, and even leukemia, further expanding the spectrum of NTRK-associated tumors. NTRK fusions are oncogenic drivers and diagnostic markers for certain cancer types that we have mentioned before. The FDA approval of THERAPEUTIc inhibitors of NTRK fusion-positive tumors is an important milestone for tumor-independent targeted therapies based on molecular profile. And last, the identification of NTRK fusions is critical in pediatric cancer care as it provides genomic evidence for tumor diagnosis and targeted therapy for patients with relapsed or unresectable tumors. Last but not the least, I would like to thank all the members from DGD Cancer Genomic Diagnostics Lab, especially Dr. Marilyn Lee, who's leading the team and also being an incredible mentor, and also Fumi Lin, who has put a lot of work in identifying a lot of the novel fusions, who will be giving another talk in a different panel on tomorrow afternoon. And also all the other DGD members and CHOP Cancer Center and Pathology Department. Thank you very much.
We do have time for questions.
Hi, very nice talk, thank you. My name is Shuri, I'm from Intermountain Precision Genomics. So I'm curious about the NTRK fusion you found in 1.6% of your cases. Did you really look into their breakpoint and orientation? Because sometimes there is some fusion which can be non-functional, so I'm just curious about the statistic number.
Yes, we do look at the breakpoint and the reads supporting the breakpoint, and we look for whether they are in frame and whether they are in the right orientation or not. And for the novel fusions or the fusions that we have seen the first time, we'll confirm them.
Yeah.
Other methods.
And the following question is about the therapy of NTRK. So we know the therapy response in patients is really great. So I'm really happy you find somebody you can give the treatment. But also, this medicine can cause the second resistant mutation generated. So I don't know, do you have any experience deal with this type of case?
Actually not yet. So the second case is actually the most recent case that actually have started to use the inhibitors. So we don't know yet, but I think the companies are trying to prepare for that.
Yeah. Thank you.
Thank you.